How does an ABG machine measure pH? pH is the negative log of hydrogen ion concentration. So the more hydrogen ions, the lower the pH. It's quite hard to directly measure concentration of hydrogen ions, so we try and measure something else instead that then allows us to deduce the hydrogen ion concentration. Luckily, hydrogen ions are charged, so we can do something with electricity and voltage. So what we're going to do is we're going to measure a voltage, and then we're going to deduce from that voltage the concentration of hydrogen ions that are causing it. So we need some sort of circuit with a voltmeter and some electrodes. And we have a reading electrode, which is our testing electrode, and a reference electrode, which is there to complete the circuit. And you need to know that the reading electrode is generally a silver electrode with a silver chloride tip, and the reference electrode is either silver or mercury. It doesn't matter too much, and it varies depending on the model. Now, we can't just bung this into our blood sample and expect it to generate a useful number, because all it's going to do is equalize the H+ concentration across the whole circuit, and we won't get a voltage. What we need is an interface that separates a known hydrogen concentration from the unknown, or the blood sample. And to do this, we use ion-sensitive glass. This glass surrounds the reading electrode, and inside the glass is a buffer solution. Then we stick the whole thing into our blood sample. And here's the idea. At this membrane of ion-sensitive glass, which is a very special silicate structure that allows hydrogen ions to bind to the surface of the glass. It's a very thin membrane, and it separates the blood sample from a known buffer that is inside the electrode. And the simple mechanism is you're going to have hydrogen ions binding on both sides of the glass. And the glass is very, very thin. You're going to have a difference in hydrogen ion concentration between the blood and the buffer. And the bigger that difference, the greater the voltage that is going to be generated. And that is the mechanism of the whole thing. All of the rest of this is just to complete the circuit and allow the voltmeter to read what's going on across this membrane. This reference electrode is in its own buffer, but that's allowed to equilibrate with the blood, and it's only there to complete this circuit. All the magic happens at this ion-sensitive glass membrane. The basic principle is this. If you take two different metals and touch them together, they're going to handle electrons differently in terms of how easily those electrons are pulled out of the lattice and allowed to move around, which means one of those metals will hold on to electrons a little bit more than the other one. And so you will actually get a slight voltage and electron movement from one metal to another. You will get a voltage generated just by touching two different metals together. It's a tiny, tiny voltage. It's not going to do any useful electrical work, but it exists. And the same principle happens with liquids. However, you can't keep liquids separate unless you have a very thin membrane, and that's what the glass does. It allows these two liquids to essentially touch in the same manner that these metals are touching, but it doesn't allow them to mix and equilibrate with each other. So you're simply measuring the hydrogen ion concentration difference between these two samples by measuring the voltage that is generated across that glass membrane, and the rest of the circuitry just completes this circuit and allows that voltage to be measured. You then calibrate that voltage because we know the behavior of this electrode, we know the behavior of this buffer solution. The only bit we don't know is the difference between here and here. So the rest of these readings can all be calibrated out, and the one that's left behind is the one we're interested in. And then you simply calibrate it to known pH samples, and then you can measure the voltage of your blood sample, and the machine will tell you what the H+ concentration is.